How's everyone doing?
Good, thank you. What about you?
I'm doing okay. It was a scramble this morning to get ready on time. But here I am with my wet hair and all. Okay, so today I am going to just do a good old-fashioned lecture because this material is so important to your understanding for the whole rest of the semester. I really just want to hit the highlights of the 4 different categories of macromolecules. And then at the very end, as promised in the email, if you are wanting to change lab partners, I will give you the opportunity to stay online and talk to other people who want to change lab partners also. And if you can work out a new arrangement, that's great. I just can't figure out a way to execute that myself. So I'm going to allow you to do that. But I also just wanted to do a quick reality check of how many labs we really have left that you need to work with lab partners for. I will just say that right now, you know, one of the big advantages to having lab partners is if you did have something that spilled in your lab kits, we're trying to get those materials replaced. But if for some reason we can't, then it obviously benefits you to have a lab partner who can do that part of the experiment for you and you guys can share data. So if you're thinking you just want to work on your own, I would encourage you to rethink that just simply based on that reason, not to mention the fact that you benefit tremendously from being able to work together. Okay, that's it about lab partners for now. Okay, so this week we are talking about macromolecules, and I just want to list the 4 categories of macromolecule on the board for you. So carbohydrates. Okay, I already know I need a better black marker here. Okay, that's better. Lipids, which of course includes the fats, but it includes some other molecules that we don't necessarily consider fats in our diet. It includes proteins and nucleic acids. So most of your body is made up of these 4 molecules, obviously plus water. And so what your cell is doing on a regular basis involves these 4 macromolecules. So it's very important to understand them, understand the differences between them, and just some of the major features that you would associate with each of those. You also need to be able to visually recognize them. So I'm going to talk about that a little bit today too. Proteins and nucleic acids are so connected that you really can't talk about one without the other. They are very different in structure and function, but their functions go together. So we'll talk about that too. So just real quickly, I want to do just a quick screen share. Oops. Sorry, it's reluctant to let me do that right now. Sorry, I've got to go back to home. Okay, can you all see just the basic Canvas page?
Yes.
So for week 5, we're just looking at carbohydrates and lipids. Okay, so next week, you're going to be looking at proteins and nucleic acids. So I don't want to get too far ahead and review proteins and nucleic acids today, but I will draw their structure today because I would like you to start out with just a picture of all 4 together so you can compare them. And we're going to be using the names of the functional groups to identify those macromolecules. We won't talk about the properties of proteins and nucleic acids today though. You're doing that next week. So this week you're just watching really three lecture videos. And again, you know, no exam this week. I encourage you to look at the exam-free study guide if you haven't already. I think it's good to watch those videos with the questions in mind so you really know what to focus on in the videos. Okay, I'm going to stop sharing my screen then so you can just look at the board. Okay, so let's start with carbohydrates. And I'm going to just draw the basic structure of a carbohydrate. And realize that when sugars are in solution, which you are, a big bag of solution, you are mostly water. So you would never have a sugar in your body that wasn't in solution unless it was a structural polysaccharide, which we really don't have in our bodies, but plants do, fungi do. So if we have a carbohydrate in our bodies, it's going to be a ring structure. I don't like drawing the ring structure on the board though, because it's hard to compare that with the other macromolecules. So I'm going to draw the linear structure of a carbohydrate. Remember that all organic molecules are a chain of carbon. So we're going to start out with just a chain of carbon. I really don't like that glare on the screen. I'm going to turn off that light really fast. I'm sorry, I'll be right back. I don't know if that took care of the problem or not. It's a little better. Okay, and then what makes you able to identify this as a carbohydrate is first, we're going to have multiples of this functional group. Does anyone remember what that functional group is called?
Carbonyl.
It's just an oxygen and hydrogen together.
Hydroxyl.
Hydroxyl, yes. So multiple hydroxyl groups. So that's one of the hallmarks of a carbohydrate or sugar. And then, as you guessed a minute ago, here's our carbonyl group. So carbonyl group is going to be the other clue that this is a sugar. Sorry, that's a Y. Not very clearly. And really the part of this is the carbonyl group isn't the hydrogen, it's this whole thing together, but it's really primarily this. Because that could be in the middle of the molecule. So you could have a carbonyl group anywhere in this molecule and it would be a sugar. Some sugars have the carbonyl group at the end and some have it in the middle, and then you wouldn't have that OH there. Okay, so this is what makes it a sugar. Carbonyl group and multiple hydroxyl groups. The molecule this looks the most similar to is a lipid. So where most students get confused is telling a lipid from a carbohydrate. If this was a lipid, it wouldn't have any hydroxyl groups. Instead, it would be all carbons and hydrogens, which we call a hydrocarbon chain, and it would have a carboxyl group here instead. I don't think I can fit that on the board with this. So I'm going to have to erase this to draw the other one. I apologize for that. I wish I had a bigger board. Okay, so this is a carbohydrate. This would be a monosaccharide. This is a simple sugar. Again, multiple hydroxyl groups and a carbonyl group. On the exam, you would never have to draw this. You need to be able to visually recognize it. Okay, a lipid, I'm going to draw a fatty acid, which is one of the categories of lipids. Boy, I'm just really not writing very well today. So I'm going to do 6 carbons again, just so you can really compare the two. In reality, a fatty acid chain would be hundreds of carbons. It wouldn't just be 6. Okay, the first thing that's different is there are no hydroxyl groups. Instead, this is just a hydrocarbon chain, which means it's just carbon and hydrogen. So hydrocarbon chain means just a chain of carbon and hydrogen. And this is already what makes this molecule non-polar. or hydrophobic. This hydrocarbon chain is nonpolar. It doesn't mix with water. And that's why lipids don't mix with water. Okay, compared to the sugar, hydroxyl groups are very polar. And you already know that sugar mixes with water because we do that in our everyday lives. So sugars are polar and lipids are nonpolar. So just that slight difference in structure, having those hydroxyl groups versus just a hydrocarbon chain makes a huge difference in the chemistry of that molecule. Then the other difference is at the terminus of this molecule, rather than having a carbonyl group, it's going to have a carboxyl group. And remember, a carboxyl group is kind of a combination of carbonyl and hydroxyl. So you have an OH here and a carbon double bonded to an oxygen. So this part right here is our other clue that we're looking at a lipid, because we have a carboxyl group. You're going to see a carboxyl group when we look at proteins also, but proteins don't have this hydrocarbon chain. So those are the two that most students get confused. The protein and the nucleic acid are going to look really different. And before we even get into talking about properties of carbohydrates and lipids, I just want to quickly draw the protein and the nucleic acid. But before we do that, does anyone have any questions? You can either ask verbally or you can put it in the chat. Either way, I'll turn on my chat.
I have a question.
Sure.
So for the lipids, does it have to have both the hydrocarbon chain and the carboxyl group, or is it one or the other? Is it always both?
It's always both. That's a good question. It's always both. Yeah, great question. Anybody else with questions about the structure? Okay, I'm just going to quickly then draw a protein and a nucleic acid. And for both the protein and the nucleic acid, I'm just going to draw the monomer, not the polymer, because obviously I have this much board space. All right, so kind of limited. So I drew a monomer for the carbohydrate. I'm going to draw a monomer for the protein and the nucleic acid as well. Remember, lipids don't have monomers and polymers. They don't follow that pattern. Okay, so for protein, I'm just going to draw the monomer. I really just need to drop that on the ground, so I'll quit using it. So this is going to be the basic amino acid structure. So amino acids are the monomers of proteins, and we have 20 different amino acids. Okay, so they're all different in structure, but they all have the basis of their structure that's identical. And that is they all have a central carbon. It's often called the alpha carbon, but you don't need to know that. Just know there's a carbon in the center. And then this functional group, which we just saw, what's this functional group called?
Carboxyl.
Carboxyl, exactly. This is a carboxyl group. Okay, now this is how you're going to identify this as being a protein, this functional group. Does anyone remember what this functional group is called?
Amino.
Exactly. This is called the amino group. Okay, difference between an amino acid and an amino group. Amino group is a functional group that consists of a nitrogen and either 2 or 3 hydrogens, depending on if this is ionized or not. So if you see this nitrogen with either 2 or 3 hydrogens attached, that is an amino group. This entire molecule is going to be an amino acid. Difference in those two. Okay, then there's a hydrogen. And remember, carbon has to share 4 pairs of electrons. So 1, 2, 3, that means we need something else here. And this is where all 20 amino acids differ in structure. This is called the R group. And the R group is where all 20 amino acids differ in structure. It can be as simple as just another hydrogen, or it can be something very complex. But all 20 have a different R group. We have 20 different amino acids that make up our proteins. A protein has to have a very specific amino acid sequence. It's really one of the most important things you can learn in this class. Every single protein in your body has to have a specific amino acid sequence, and that amino acid sequence is the same for every human on Earth. For that protein to function correctly and have the correct shape, it has to have the correct sequence of amino acids. The basis for every single human genetic disorder is having a protein that doesn't function correctly because it has the wrong amino acid sequence. And we'll talk about how that happens too. But the sequence of amino acids in a protein is what ultimately causes the shape. And those 20 different amino acids have to be in a specific order, depending on what protein you're talking about. So if you're talking about lactase enzyme, which we've already talked about a couple of times now, lactase enzyme for every single human on Earth must have the exact same sequence of amino acids to function correctly and have the correct shape. So that's going to be a big deal for the whole rest of the semester in this class. This is the basic structure, and when you listen to the protein lecture, you'll see a slide that shows the structure of all 20 amino acids. For now, you just need to know this basic structure. You can easily tell this apart from the other 3 categories, lipids, carbohydrates, and nucleic acids, because of this amino group. It's the only one that will have the amino group. Are there any questions about the amino acid structure? What we call a protein then is a chain of these. Okay, you would never have a protein that was just one amino acid. Okay, then lastly, the 4th category of macromolecule that you need to be able to visually recognize and know about are nucleic acids. And again, I'm going to draw the monomer, which is called a nucleotide. So the polymer would be the chain of those called a polynucleotide. But I'm just going to draw the monomer. Okay, we start with a 5-carbon sugar called a pentose sugar. So a pentagram has 5 sides. Each one of those sides represents a carbon. So this is a 5-carbon sugar. And depending on which nucleic acid you were talking about, it's really just one of 2 pentose sugars. It's either what we call ribose, which you will see the structure of when you watch the carbohydrates video. So interesting, nucleic acids are made partially of a sugar. So within this category of macromolecule, you have lurking at the center another macromolecule, a sugar. But there's a lot more going on here than just this sugar. So it's either ribose or deoxyribose. And when you watch the nucleic acid video, you'll see the basic difference in structure between ribose and deoxyribose. that's not critical to you recognizing this as a nucleic acid. So I'm not going to give you that detail right now. Okay, over here, we have a functional group. Okay, so this gets a little tricky. This is phosphorus double bonded to one of the oxygens. So let me draw this a little more clearly. So this is a double bond to an oxygen. This is a single bond, a single bond, a single bond. Okay, so this phosphorus actually has 5 bonds. 1 double bond, single, single, single. And then these are actually negative signs. So this is the ionized form. This phosphorus in the middle is your clue to what functional group you're looking at. Does anyone remember what this one is called?
A phosphate group?
Phosphate group?
Exactly, this is a phosphate group. So 2 clues for recognizing this as a nucleic acid. One is this pentose sugar in the middle, and then this phosphate group. It's the only macromolecule that we'll look at that you'll visually recognize as having this phosphate group. Now, some nucleic acids actually have 2 of these phosphate groups, and some actually have 3 of these. And you'll see that when we talk about nucleic acids in more detail. And then over here, we have what's called a nitrogenous base. It's a base that has nitrogen as part of it. I'm going to write that more clearly. It's kind of hard to see there. I'm going to write the word first, and then I'll draw the circle around it. So nitrogenous means it has nitrogen as part of it. And it's really going to be one of 4 different bases, depending on if we're looking at DNA or RNA or ATP or ADP. And you'll learn that later too, but this will be some other molecule here. So just like with the amino acid, how we just put an R group for now to represent what we're going to have attached here. Right now, we're just calling this a base that has nitrogen as part of it. And you'll see what that looks like in more detail. But again, the way you're really going to recognize this as being a nucleic acid, the most basic ways are this pentose sugar with at least 1 phosphate group. And it could have 2 or 3 depending on which one we're looking at. So this looks really, really different than the other 3. The nucleic acid is really, I think, the most unique in structure and easiest to recognize. And then the amino acid, again, the way you're going to recognize that is the amino group. It does have a carboxyl group, which gets a little confusing, but it doesn't have that long hydrocarbon chain, so you wouldn't confuse it with a lipid. The lipid and the carbohydrate are really the 2 that look the most similar. So you would only have to recognize those. You wouldn't have to draw them, but you will have to, in a multiple choice question, name the macromolecule on the next exam. Any other questions about just the basic structure? Okay, I want to talk a little bit about carbohydrates. A lot of this is reviewed from the video, but I just think it's so important. I want to make sure everyone's on the same page. So when we're talking about carbohydrates, the monomers, which again are molecules all on their own, but those are the individual subunits that are going to link together to form the polymer. The monomers are called monosaccharides. And the polymers are called polysaccharides. And in the lecture video, you have examples of monosaccharides and polysaccharides that you need to know about. And then the sugars are kind of unique in that we have a really simple polymer that's really just 2 monosaccharides bonded together. and that's called a disaccharide. It's really the only category of macromolecule in which you can have two linked together. It's technically a polymer, but usually when we hear the word polymer, we think of this big long chain of monomers. But in sugars, we actually have disaccharides that are biologically significant. And the three that you're given that you need to know are sucrose, maltose, and lactose. So here's lactose again. We've talked about lactose a lot now. So sucrose is two monosaccharides linked together, and those two monosaccharides are glucose and fructose. And remember, glucose and fructose are what we call structural isomers. They have the exact same chemical formula, but different structure. They're both C6H12O6, so different arrangement of those same exact atoms. Maltose is just two glucose molecules linked together. So glucose plus glucose. My elbow's hitting my desk when I'm trying to write. It's really inhibiting my writing skills right now. Okay, and then lactose you already know from the natural selection talk, that is glucose plus galactose. So galactose is milk sugar. Lactose is milk sugar. So sugars are a little unique again in that they have this molecule that's biologically significant that's just two of the monomers linked together. So glucose, fructose, galactose are all structural isomers. They're all three C6H12O6. And that's the general chemical formula ratio for any monosaccharide. All monosaccharides have the general formula of having twice as many hydrogens as carbons and oxygens. So it's often written as CH2O times some number. So you have equal number of carbon and oxygen and then twice as many hydrogens. So this is monosaccharide. And again, you would never have to draw any of these. You would just have to be able to visually recognize that it's a carbohydrate. You don't need to recognize the difference in structure between glucose, fructose, and galactose. Just realize they all three have the exact same chemical formula. And then the other two monosaccharides that you really need to know about are ribose and deoxyribose, because those make up nucleic acids that we're going to be talking about too. So in the video, you saw that the other monosaccharides that you need to know about are smaller sugars, they're the pentose sugars. So ribose and deoxyribose. These are also monosaccharides. Saccharide's an interesting word. It has two C's together from our French root. I always say the French have a lot of extra letters in their words. Don't really get pronounced. Okay, any questions about that? We obviously mostly eat polysaccharides. Oh, Martel, did you have a question?
Yeah, real quick. The ribose and deoxyribose, where do those derive from?
That's a good question. So these are going to be these are the sugars that make up the nucleic acids. So ribose is found in RNA.
And then deoxyribose is part of our DNA, right?
Exactly, yeah. And really the only difference, so deoxy means without oxygen. And if you look at those two sugars, It's a great question.
So it's to break it down, it's just, I don't know, genetic sugar. I'm trying to put this to. I'm trying to process it in my brain. You know.
Yeah, that's a good question. So it's just that sugar is just part of the nucleic acid molecule, but ribose is also found in ATP, which is the energy currency of the cell, and ADP, which ATP breaks down into. So it's interesting that the sugar that makes up our nucleic acids also, you know, as part of our genome, is also a nucleic acid that becomes an energy source for our cells. I mean, those molecules are so similar in structure. This stands for adenosine triphosphate. So it actually has 3 phosphate groups, and adenosine diphosphate actually has 2 phosphate groups. So you're going to see, if this was RNA, it would have 1 phosphate group. ADP is going to have 2, and ATP is going to have 3 in that location there. So they're really similar in structure. So deoxyribose is missing an oxygen right there. Otherwise, it's identical in structure to ribose. It's just missing that oxygen right there. So this makes up our DNA, and this makes up our RNA, our ADP, our ATP.
ATP.
And it'll make more sense when you guys watch the nucleic acid video. I hesitated to even introduce this now. I just really like to show you the structures of all 4 at once so you can really compare them. Paris asks, why are the points on that 5-point shape origination? They are carbon. So that's a good question. So these are carbons, but the short form for drawing the sugar, you just don't draw the carbons. So when you really look at the detailed structure of those sugars, you'll see the carbons there. Let me show you something really fast. Sorry, I'll go back to. I'm going to go back to a semester when I was allowed to post PowerPoint slides. So that is not this semester. It is no longer allowed. So I'm letting you into the secret archives here. This is when we were face to face. Oh goodness, there are a lot of people using the internet right now in my house. My daughter sadly started UCLA graduate school online this week, and it breaks my heart because the best thing about graduate school is that camaraderie you build with your classmates and getting that relationship with your professors that's kind of on a different level than being an undergrad. And, you know, being on the UCLA campus, how great would that be? Instead, she's in her bedroom in Poway. Really bums me out. And then we have a son doing his senior year online, and another one doing his first semester at Palomar online. Okay, so when we draw the ring structure of a sugar, so this would be glucose in its linear form. So there are the 6 carbons. Here's the carbonyl group. Here are the multiple hydroxyl groups. When this is in solution, it forms a ring that looks like this. You guys would want to drive to my house and hit me in the head with something if I started trying to draw that on the board every time. So I don't do that. The short version is just to draw the ring. So when we're talking about ribose and deoxyribose, let me get to the sugars here. polysaccharides. So this is ribose in its linear form. So when it forms a ring, we don't draw all those carbons on the ring. So just as the formula is CH2O times some number for all monosaccharides, in this case, it's a 5-carbon sugar, so that number is going to be 5. So that means for ribose, the formula is C5H10O5. So twice as many hydrogens, equal numbers of carbons and oxygens. Now DNA, that sugar is deoxyribose. It's going to have a slightly different formula because it's missing an oxygen, so it doesn't really follow this formula. It would actually be C5H10O4 because it's missing an oxygen. Deoxyribose without oxygen. It's missing that one oxygen.
Is there any way that I could see the board?
Yes, let me do a stop share.
Thank you.
Great questions today, everyone. Thank you so much for asking questions. Okay, we mostly eat the polymer, polysaccharides, and then we break that down because it has to be the size of a monosaccharide to get into your cells. So disaccharides cannot get into your cells. Polysaccharides of any size can't get into your cells. Only monosaccharides can. So we have to eventually break those polymers down into the monomer to get them into our cells, use them for energy or whatever else we need them for. But we mostly eat the polymer. I mean, most of us aren't drinking glucose all day. So the polymers of carbohydrates are the polysaccharides. And we have a few that are biologically significant. We have starch. And really, these are all a chain of glucose. Every single one of these is a chain of glucose. Starch is a chain of glucose. We talk about 2 kinds of starch in the video, and so I'll come back to starch and give you some more details on starch. Glycogen, cellulose, and then a structural polysaccharide called chitin. It's spelled like chitin, but it's chitin.
Okay.
Starch is how plants store their sugar. This is the long-term storage form of sugars for plants. So whenever we eat starch, we are eating a plant product, and that plant has stored sugar in the form of starch. It's a long chain of glucose. So this is carb storage in plants. And this is the main form of sugar that we eat. As you know, some plant products are higher in starch than others. So if you eat a potato, that's almost pure starch. That is a structure that that plant uses specifically for storing carbohydrates. And if you've ever seen a potato growing, I know today's trash day at my house. So this morning I got up and went through our produce basket and threw out all the sprouting sweet potatoes that have been sitting there for several weeks and nobody ate. And they all have these big old plants coming out of them. So as you know, the main part of the plant for a potato isn't the potato, right? That's the part that's underground, and it's called actually a tuber, and that's where that plant is storing starch. So when you eat that part of the plant, you're just getting pure starch pretty much. Carrots, that's a lot of carbohydrate. That's a storage structure for that plant. And then as you know, there are parts of the plant you can eat, like kale. Kale hardly has any starch in it. Kale is high in what's called cellulose that makes up the cell walls of plants. So anytime you have a plant that's, you know, kind of hard to chew, a little more what we call fibrous, it's because of the cellulose that makes up the cell walls. And as you'll see in the videos, if you haven't watched them yet, we cannot digest cellulose. We cannot break it down into glucose. So animals that only eat plants have to have bacteria in their guts that break down the cellulose for them. No animals on Earth have the enzymes to break down cellulose into glucose. So what's crazy is starch and cellulose, both found in plants. The cellulose is on the very outside of the cells. Okay, so cell wall, you're gonna learn a lot about cell walls coming up in the cell lecture. This is the cell, boy, that is the worst green marker ever. This is the cell wall of the plants, and it's around the plasma membrane. So that plasma membrane is mostly made of phospholipids, whereas that cell wall is made of carbohydrate. And it's really just a chain of glucose, but what happens is the bond holding the glucose molecules together is just a little different in structure, and we don't have enzymes to digest this. And then what will happen is inside here, if this is a plant cell, inside here there's going to be a bunch of starch. We can digest this starch, we cannot digest that cell wall. But animals that eat a lot of cellulose and have to get that glucose out of the plant, they have bacteria in their guts. Bacteria digest cellulose. Animals do not have the enzymes to do that. So we don't have those bacteria in our guts. You have, if you're a hoofed mammal, everybody who has hooves has the enzymes in the form of bacteria. So the animal itself doesn't make those enzymes, but it has symbiotic bacteria that live in a section of their gut called the rumen. And those bacteria are who digest that for them. So if you think about an elephant, you can build a whole elephant just on the glucose that they get out of the cell walls of the plants they eat. And they can't digest that. It's the bacteria that do that for them. It's one of the many beneficial roles of bacteria. Gut bacteria are important for every animal, but especially if you're a hooved animal. You're a hooved mammal, you 100% rely on those bacteria to digest your food for you. It's pretty crazy. Glycogen is how we store carbohydrates. And that's a hugely important story in the videos, and you can guarantee an essay question on exam 3 that talks about blood sugar regulation. So I just want to spend a couple of minutes today going through that story. I know it's in the video, but it's so hugely important, and there are a few major points in that video that are important. But before I move on, while we're on this list, before I explain really the role of glycogen in our bodies, I just want to talk for a second about chitin, because if you go on to take microbiology, you will learn about fungi. Fungi make up their cell walls with chitin. Their cell walls are not made of cellulose, it's made of chitin. Also, arthropods, so insects, crustaceans, arachnids, ticks and mites and spiders and crabs and, you know, all the arthropods, their skeleton is on the outside of their body. It's called an exoskeleton, and it's made partially of chitin. We make dissolving surgical thread from insect exoskeletons. So some people, you know, when they get those dissolving stitches, they're actually allergic to them. They start really itching and they'll get a rash right away. It's because there's something in the insect that their body is responding to. But chitin is a structural polysaccharide that actually has nitrogen as part of it. But you'll talk about fungi a lot in microbiology. Fungi are a major pathogen of humans. So they're really important to understand. And when we talk about human pathogens at the end of the semester, we'll learn about fungi. I actually listened to a podcast yesterday about fungi and how they're becoming more and more of a health concern to humans because they're very, very hard to kill. And part of the reason they're really hard to kill is they are really metabolically similar to us. They have a lot of enzymes that are the same as human enzymes. So to kill fungi, you have to really poison yourself and really interfere with your own enzyme activity. So that's why if you've ever taken any antifungal medications, a lot of times they have to do liver testing on you because it's so damaging to your liver because it's interfering with your key enzymes in order to kill the fungal infection. So we will talk more about fungi, but important to realize that chitin is an important part of their cell walls, and it is a structural polysaccharide, different in structure than cellulose, which makes up the cell walls of plants. Cellulose, of course, becomes fiber in our diets, and I talk about that in the video. Any questions just about this basic list before I talk about blood sugar regulation and the role of glycogen? Also, just to let you know, now that we're kind of into the meat of the semester, I will be doing more of this in our lecture discussions. Just really a lot of review. Obviously, it doesn't take the place of watching the videos. Because I'm really condensing 3 or 4 lectures into one. I'm just giving you the highlights. Okay, let's talk about blood sugar regulation. This really affects our health as Americans. So I've loved reading your diet posts over the past week. Reminder, your post is due today. So you need to make your diet post by today, and you need to respond to 2 other posts by Friday. And by the way, I've loved the responses I have read so far. If you have made responses so far, thank you. You're really making some great responses to other students' posts. I really appreciate the efforts you all are putting forth. The diets are really interesting. At the heart of many of our health problems in the United States is that we do a really poor job of regulating what kinds of carbohydrates we eat. Now, carbohydrates are by far the most important thing you put in your body every day. If you are not eating enough carbohydrates, I don't care what any fad diet tells you, you are damaging your body if you're not eating enough carbohydrates, because you have to have carbohydrates to make ATP. We will talk a lot about cellular respiration this semester. Every single chemical reaction in your body that requires energy, which is almost all of them, must have ATP to power that reaction. We make ATP from carbohydrates through cellular respiration. So we take glucose and oxygen, and we go through many, many steps. This arrow represents a lot of steps that you will learn the details of. You'll learn exactly what that arrow represents. And we end up making something very different from glucose and oxygen. From those same atoms, we make water and CO2, and we make on average 36 to 38 ATP from that one glucose. That ATP, that is energy. That is the energy that you need to use to drive reactions in the cell. We don't have the ability as living organisms to just take glucose and power our cells. Okay, so different from your car engine, you put fuel in your car, and your car breaks those covalent bonds of the fuel molecule and directly uses that released energy to power the engine of your car. To power our engine, we do something similar. We break the bonds of glucose and we release that energy, but we then package that energy in the form of ATP. And that's pretty cool, because what that means is we don't have to be on a glucose drip all day. So if you run out of gas in your car, if you don't have you know, an electric engine or a battery like in a hybrid, once you run out of fuel, that engine doesn't run anymore. In the case of us, we don't have to have a constant fuel source, because what we can do is, number one, we can store this as glycogen, and we're going to talk about how we do that. So we have a little bit of reserve sugar hanging around. And also we convert that energy in the sugar molecule to ATP. And we can now use that anytime, any place in the body. And what's crazy is you make and use your body weight in ATP every day. And if you're not eating enough sugars to fuel production of ATP, your body will start breaking down muscles and organs and use those proteins and fats to make sugars that can then be used to make ATP. So a constant supply of healthy carbohydrates, and we're going to talk about what that means, is imperative to your normal function. And I will tell you that the biochemistry of the human body hasn't magically changed in the past 20 years. Okay, we have the same biochemistry we have always had. And so any diet that tells you otherwise is just false. The chemistry of the human body is not someone's opinion. It is a fact. And so when you're fueling your body with something like a protein or a fat instead, there is going to be a price to pay. And yes, you will lose weight, but it comes at a cost to your body over time. So wait, the diet I did is power protein diet and reduce the amount of carbs you eat. Yes. Okay. I will tell you that since COVID started, okay, so I'm 100% guilty. So do what I say, not what I do. Okay. Okay, I'm 55 years old and I had gained too much weight. I had a physical in January and my doctor told me, you have to lose at least 10 pounds. So I've tried for years, I've tried to lose weight and I've always eaten really healthy, but my lifestyle is my husband and I love making dinner together. And while we're making dinner, we love our cheese plate with our bread and we love our wine and we really love beer. And so before I knew it, I'd put on 25 pounds.
It's the beer that gets you.
Yeah, the beer. It's evil. It's so good.
Straight to the stomach.
Yeah, well, alcohol calories are tough to burn. But, you know, and honestly, the reason you get a beer belly is because your body is trying to protect your organs from the alcohol. So you're accumulating fat in your midsection to protect your organs. It's like a defense mechanism. That's why anyone who drinks a lot of alcohol, you don't store fat evenly through your whole body. It's differentially stored around your organs to kind of protect your organs.
Doesn't alcohol turn straight into fat?
For the most part, yes, because it's too hard to burn, and that's our long-term storage. So any carbs you don't use right away get stored as fat. They go into long-term storage, which is fat. So yeah, they get converted to fat more readily than these other carbs. But so I've been doing the low carb thing because I didn't have a choice, and it works. I mean, it definitely works. I've lost 22 pounds since January on low carb. But
Nice.
Congratulations.
Thank you. I've gone down 2 sizes in clothes. It's awesome. I mean, one thing is I'm walking a lot, right? Because what else do I have to do? I walk in our houses. But also the low carb thing, it does work, but it is really damaging to your body. And I'm going to tell you why it's damaging to your body. You can do it short term, but you shouldn't do it long term because it can really mess you up. But this is why you need carbs. So I just wanted to start off the carb lecture saying, you know, carbs get a bad rap these days, but it's because we as Americans, we abuse carbs, we overeat carbs, and we eat the wrong kind of carbs. And it just goes to fat storage. So difference in quality of the kinds of carbs you eat. So 2 kinds of starch. So we're mostly eating starch. We have what's called amylose, which we call simple starch. And it's just a simple chain of glucose. That's why it's called simple starch. It's just really simple in structure. And then we have what's called amylopectin. And this is what we call complex starch. So often called complex carbohydrates. This is really the best thing you can eat. And we'll talk about why in terms of blood sugar regulation and how it affects your weight. There are pictures of this. I could just keep switching to screen view, but you guys will see the structure of these two starches when you watch the videos if you haven't already. This one's called complex because it has some side branches. So if this is our chain of glucose, amylopectin has, you know, at least one side branch. And what this means is this is just a simple swirly chain of glucose. This one is digested by different enzymes than this one. In fact, this is digested by enzymes starting in your mouth. In your saliva, you have an enzyme called amylase. Okay, again, love these enzyme names. They tell you what they do and they end in ASE. So amylase breaks down amylose, and it's in your saliva. So when you eat this simple starch, it actually starts breaking down in your mouth. So by the time you've swallowed it, you've already released that glucose and it's going to go straight into your bloodstream. So this is all the stuff that tastes good. You know, this is the baguette.
The French fries and the potato chips and all that stuff.
Yeah. So pasta, white rice, any kind of white bread. You know, a lot of wheat bread too. Yeah, wheat bread is mostly just regular flour, and then they throw some grains in there to make it look brown, or they just don't bleach the flour. But this gives you that instant sugar rush. And we're going to talk about how that affects your blood sugar differently in a minute. Right now, I'm just kind of laying out the difference between the two starches, and then I'm going to draw the diagram of your blood sugar regulation, and we'll think about it in terms of that. Amylopectin, it does not get digested in your mouth. In fact, it doesn't get digested until it gets to the small intestine. So this is broken down by enzymes in the small intestine. Okay, when the food is in your stomach, so you've chewed it, you've swallowed it, it's now there in your stomach. Does it all go into your small intestine at once, or is it pulsed out a little bit at a time to your small intestine?
A little bit at a time. Peristalsis.
Yep, peristalsis. It goes in a little bit at a time. And remember, the reason for that is pH. Because we can't just have all that acidic food just go into the small intestine, because it will denature all the digestive enzymes. We can never break it down and get it into our cells. For us to be able to break down this starch, a little tiny bit of food at a time comes from the stomach. We send in that buffer. So we send in the bicarbonate ions to tie up all those free hydrogen ions, get the pH back up. Now we can digest it. So the cool thing about eating amylopectin is you have a little bit at a time getting digested. So you get this slow, steady release of carbohydrates into your bloodstream. So this is going to be slow, steady glucose release into your bloodstream, okay, versus big glucose rush. This is going to kind of be a big rush all at once. So amylopectin, you know, if your blood sugar is low at first, it's going to go up and it's going to be pretty steady, kind of like this with amylopectin over a long period of time. With amylose, your blood sugar is going to spike up and go back down.
Quick question.
Sure.
So I know that you said that the process with amylose starts when you start chewing the food. The saliva breaks it down. I remember that part in anatomy. Not necessarily that it was the simple and the complex differences like that, but so it's the minute you swallow it and it reaches your stomach, that's when that process is considered over and you get the spike, or is it while you're chewing it and breaking it down in that part, the mastication process, then you get that spike?
That's a good question. And as you were asking that question, I was thinking, well, what if you just swallowed it whole? But then it wouldn't taste good, right? Like the whole reason we eat it is it tastes good. But yeah,
Somebody
you're
just
right.
swallows a whole slice of cheesecake.
They should just put it in pill form. Like we should just
take the guilt away.
So yeah, it starts in the mouth, and then whatever doesn't get digested by the time it's swallowed, it'll continue in the small intestine.
Right. So you already get that spike while you're chewing it then.
Right, exactly.
Oh,
so that's why, when you have really low blood sugar, you start feeling sluggish, you can go eat a cracker or a piece of bread or something, and you just immediately, like, your head clears. Because your brain cells are the only cells in your body that don't have to wait for insulin to get produced. So when your blood sugar is really low and you're starting to feel it, if you eat some simple carbs, then you right away get that, you can feel your head clear, because that glucose is going to go straight into your brain cells. The brain cells get the glucose first, and then everybody else kind of has to wait for insulin to come on scene for the other cells to be able to get the glucose in.
Nice.
So yeah, I guess whatever you don't digest in the mouth, you're going to not digest it again until the small intestine, because you don't get those enzymes coming into the stomach. Okay, so now you kind of know the difference between these two. Let's talk about how that affects your blood sugar regulation. So blood glucose. You have a set point for blood glucose level in your body. And it doesn't even matter what that number is. I'm not even going to give it to you. I don't like giving numbers in this class because I don't want you to think you need to memorize those numbers. If you're curious what it is, it's in the video. Okay, but this is one of the many things we have in our body that has to stay constant, because we have to constantly make ATP. So we don't quit making ATP when we go to sleep at night. We don't quit making ATP just because we haven't eaten for a few hours. So we need a way to have a constant supply of glucose so we can constantly make ATP. I'll give you an example of how important this is. The poison cyanide, which is rat poison, the way it kills an organism is it shuts down cell respiration. It shuts down the process of making ATP. The second you stop making ATP, you're dead. That's it. You have to make ATP all day or you're dead. Nothing in your cell can happen. So you immediately just die when cell respiration stops. So this is one of the many things in our body that it's important to keep it at a constant level. And that process of keeping something at a constant level is called homeostasis. So homeo means same, and stasis means state. So keeping something at the same state. We have a whole homeostatic mechanism that keeps our blood glucose at the same state. When you don't do that, you start having some serious health problems that occur. But the normal mechanism works this way. So here's our set point. And anytime you have a set point for something, like just think of your body temperature. Our body temperature stays at around 98.6, but by the way, humans are getting colder. I don't know, everywhere you go lately, you're getting your temperature taken, right? Like just driving onto campus yesterday, the thing get, temporal scanner for my temperature. And, sometimes you're surprised when they say, oh, your temperature's 96 or your temperature's 97. Humans are getting colder. There are some theories on why that's happening, but so you know, we say 98.6 just because that's what we're used to saying, right? But most of us are actually a little colder than 98.6. So if your temperature gets too high, the body's going to bring it down. If the temperature gets too low, the body's going to bring it up. Same thing here. So just in general, without me drawing into details, if your blood glucose goes up, we're going to do something to bring it back down to the set point. And if it gets too low, we're going to do something to bring it back up to the set point. And that's how homeostasis works. This is the set point. If it gets too high, we'll bring it down. If it gets too low, we'll bring it up. Kind of think of the thermostat in your house. You set your thermostat at 75 degrees. If it gets too hot, the air comes on. If it gets too cold, the heater comes on. Same thing with regulating your blood glucose. So the way we regulate it is this. So let's say you eat carbs, and we won't talk about what kind they are yet. We're just going to say in general, you're going to eat some carbs. And you start breaking that down, either in the mouth or in the intestines or both, depending on what you ate. And immediately you're going to get some type of increase in blood glucose level. Glucose is a pretty big molecule to be in your bloodstream. If you think about what's mostly in your blood, we have water, we have oxygen, we have carbon dioxide, we have some dissolved solutes. We don't have something big like glucose. Glucose is big, and it's very damaging to your blood vessels, and it can't stick around for long. So we get this glucose, which we need, okay, but we need to get it somewhere right away. It can't just be floating around in your bloodstream all day. You've all known diabetics who end up having vision problems, kidney issues, fingers and toes, or even entire, limbs sometimes that are lost to diabetes. And that's because they can't get that glucose out of their bloodstream right away. And it starts damaging all the capillaries in anywhere that has a lot of tiny capillaries. So, lots of damage to organs, but in particular, your kidneys do a lot of filtering and they have a lot of blood vessels. So that's one of the first things to be damaged when that glucose sticks around for too long. So what we do is we release a hormone called insulin. Insulin is produced by the pancreas. Pancreas does a lot of important things for us. We've already talked about several of those things. The pancreas produces bicarbonate ions. They get pumped into the small intestines to be a buffer and bring that pH back up so we can digest our food. The pancreas then produces those digestive enzymes that we need to digest our food. And the pancreas also produces two important opposing hormones that we're going to talk about in this story today, one of which is insulin. So insulin is released, and that insulin does a couple of important things. It gets glucose into your cells. So any cell that's not your brain has to have has to have insulin bound to the cell for glucose to get in. Just think of it this way. It's the key that opens the lock to get glucose into your cell. If insulin is not present in high enough amount, you can't get glucose into your cells. Oh shoot, sorry, my phone is ringing. Hold on a second, sorry. Of course, it's potential spam, because everybody knows not to call me right now. Okay, and then,
Professor?
yeah.
So what happens when we swallow a gum? Like, how does it break down?
The gum, when you swallow it,
Yeah.
you poop it out eventually.
Oh, okay.
Could take a while, kind of. I've seen scans where people have a lot of gum hanging around in various parts of the tubes. But,
Yeah. All right, thank you.
You can swallow lots of things whole that don't get digested, and they eventually come out. Okay, we've got to get that glucose out of the bloodstream. We need it in our cells to make ATP. So we're going to get some of it into our cells, but you know, most of the time we've eaten more glucose than we can get into our cells, and also we need to store a reserve. So the other thing that insulin does is it links glucose together to form a chain of glucose called glycogen. And that glycogen, that's our sugar storage short term. We store about, on average, a 4 to 6 hour supply of glycogen, and we store it mostly in our liver. Okay, and a small amount in your skeletal muscles. And if you've watched the video, you already know the answer to this, but I'll ask everyone anyway. Why do you think that mammals in general should store a little bit of glycogen in their skeletal muscles?
The
Energy use?
yeah. In case
Yeah,
that
energy
fight
for
or
what?
flight response is needed. You need to get up out of there.
Yeah, you need to escape. If you need to, you know, the whole fight or flight. Okay, not everybody's at the top of the food chain. So, you know, the squirrels who are brave enough to come into my yard with my 2 big dogs, they know the value of having a little bit of glucose stored in their skeletal muscle. They need to book it on out of there. So it's a good mechanism. It's a good strategy to have some instant energy right there at where you're going to need it, if you're going to need to get away. So when you eat a steak or any other kind of meat, you know, you're eating skeletal muscle. And so you are getting a little bit of sugar from that meat. And in fact, when you cook a piece of meat and it caramelizes on the outside, you're cooking that stored glycogen that's in that muscle. Doesn't that sound delicious? A nice cooked skeletal muscle.
Not at all.
I'm not vegetarian, but when you think of it like that, it's kind of gross. So we store about a 4 to 6 hour supply, and now insulin has either made that excess glucose get into the cells or get stored as glycogen, and now we're back down to normal. But we're constantly using that sugar, so we're not going to be at normal for long. We're going to dip down too low. And when our blood glucose gets too low, now we need to start breaking that glycogen back down and releasing it back slowly into the bloodstream as glucose. So now we've got blood glucose decreases and it's too low. And now the pancreas is going to release a different hormone called glucagon. Okay. Unfortunately, glucagon looks a lot like glycogen. It's very important to know that those are 2 different words, 2 different spellings, 2 different things. Glycogen is a chain of glucose. It's how we store glucose in our liver and a little bit in our skeletal muscles. It's the storage form of carbs in animals. Okay, glucagon is a hormone released by the pancreas, and it takes that glycogen and breaks it back down into glucose. And now that glucose is released into our bloodstream, and our blood glucose level gets back up to normal.
I'm sorry, you said that it was a hormone?
It's a hormone. Yep.
Okay.
So insulin and glucagon, both produced by 2 different types of cells in the pancreas, and they do 2 opposite things. I'm gonna go way to the top of the board real fast. Okay. So if you think of it this way, we're going between glycogen and glucose. Actually, you know what? I'm gonna put it at the bottom here. I'll erase this one little part. I'm just gonna erase this part right here for a second. So glucose This is the monomer that is going to get into our cells. Okay, glycogen. This is the storage form, and it's a chain of this. You know, this is a very simple version of it. It's got branches off the side and everything. I'm going to put my reversible arrows there. Glucose getting stored as glycogen. That's the job of insulin. Okay, so insulin's going to take us this direction. And then going the other direction, glycogen getting broken back down into glucose. That's the job of glucagon. This red has to get going here. He's a new red. Glucagon. These are called opposing hormones. They have opposite effects. I can guarantee a short answer question asking you to explain the difference in roles between insulin and glucagon. So remember, insulin actually does two important things. It gets glucose into our cells, and then it takes that excess glucose and stores it as glycogen, links it together and stores it as glycogen. Glucagon is going to do the opposite. It's going to break that glycogen back down into glucose. So one of two things can happen. Either we release all of our glucose before we eat again, and as soon as this glucose level is dipping so low and we don't have any glycogen left, we're going to release some hormones that are going to cause us to feel hungry. So in a normal person with a normal hormone balance for hunger, you're usually going to start feeling hungry when you're out of glycogen and you're not making glucose anymore. That's when you start feeling hungry again. That's when you start getting hangry. You know, you start getting low blood sugar. Your brain starts getting a little funky because you don't have enough. And that's when you'll start feeling hungry again. If you eat before that happens, so you eat more carbs when you already have some still stored, you're going to send those carbs to long-term storage. Okay, so a pretty common question, it's pretty logical, is, okay, if all I'm eating is carbs, how do I get fat from that? The way you get fat is because the body's long-term storage is to convert carbohydrates to fats. And fat is very energy dense and a good form of long-term energy storage in case you need it. So anything that is eaten in excess of this normal cycle will get stored as fat. So if you think about the difference between the two kinds of sugar, if you're eating simple carbohydrates and you're getting this big spike in blood sugar, you're going to also have a big spike in insulin getting released. And remember, this insulin has to bind to your cells, everything but your brain cells, to get glucose into your cells. And over time, if you're doing this a lot, your cells become what's called desensitized. So now a normal amount of insulin is not recognized by your cells, and you can't get that glucose into your cells. You now need a higher amount of insulin than normal. So yeah, so that's right, Martel, that's what causes that crash feeling afterwards, for sure. And that's why sometimes you get that sugar headache too, is just that big rush of sugar all at once. You get that, you start feeling a little funky from all that sugar. Okay, so type 2 diabetes is, you know, there are genetic factors that can make you more at risk of developing type 2 diabetes when you get older. But type 2 diabetes is a huge health problem in the United States because we abuse carbs. We eat too many, it's too readily available to us. Okay, so we as primitive peoples we had a hard time finding carbs. And we need a constant supply. So we crave them. We crave carbohydrates because it used to be hard to find, and that craving stimulates you to go look for it so that you'll eat it and give your body what it needs. But now it's too easy. Like, it surrounds us. It's too easy to find all at once. And so now we abuse it. And if you just bombard your body with all this simple sugar and you're releasing too much insulin all day in response to that, over time, your cells don't recognize it anymore. And now you need a higher than normal amount of insulin to be able to get that glucose into your cells. And that's called type 2 diabetes. Type 1 diabetes is not the same. Type 1 diabetes is an autoimmune disorder in which your white blood cells attack and kill cells of the pancreas that produce insulin, and you just cannot produce enough insulin. And usually by about age 5, it becomes a major health problem, but you're born with that. You're born with type 1 diabetes. Type 2 diabetes is, again, there are some genetic factors that can make you more likely to get it, but even if you have those genetic factors, you have to have the perfect storm. You have to not exercise, eat poorly, and be genetically predisposed. Even if you have the genetic factors, if you eat healthy and you exercise, you're most likely not going to develop it. And if you do, it won't be till you're like 95. Okay, so this is a major health concern, and it's 100% related to our lifestyle as Americans. We don't exercise enough because exercising your skeletal muscles pulls glucose out of your bloodstream at a very fast rate. So especially when you get to the anaerobic phase of exercise, which we'll talk about what that means when we talk about cell respiration. When you get to the point where you're low oxygen in your muscles, you start burning glucose at an accelerated rate. And that's a really good way to control that. Okay, so type 2 diabetes, mostly the result of poor diet. Think of that complex starch in comparison. If you're eating complex carbohydrates, instead of getting this big spike, you're getting this slow, gradual release of glucose into your bloodstream, slow, gradual production of insulin. That's the perfect scenario. So if you're eating stuff like brown rice, complex grains in your bread, whole grain pasta, fruits and vegetables that have those complex carbs that are harder to break down, you're going to get that more slow, steady release of sugar into the bloodstream, and that's really what you want. Because then you get slow, steady release of insulin, you're not bombarding your cells, and you're helping your body do this, because we need this constant blood glucose level. What happens at night while you're sleeping is obviously you're burning glucose at a much slower rate because you're asleep. You're not exercising, you're not pulling glucose out of your bloodstream by exercising your skeletal muscles. So your glucose needs at night are just lower, and you have some stored. So type 2 diabetes is when you eat too many simple carbs and your body becomes desensitized and you need more insulin than normal. Yes, it's becoming desensitized to the insulin. So it's kind of like if you live on a busy street, when you first move in to that house, you hear the traffic all day long and it drives you crazy. Over time, you become desensitized. You stop hearing it unless like a big loud motorcycle goes by. Okay, if you have type 2 diabetes, you live on the busy street, you're seeing insulin all day, every day, and now you have to have the loud motorcycle to be able to recognize it. So you need a higher than normal amount of insulin to get that. So, you know, most type 2 diabetics luckily are not insulin dependent yet, but it does get to that point. Most type 2 diabetics can control their diabetes with diet and exercise. Even just something simple like while they're watching TV, they have 2 little dumbbells and they're moving them. You know, just even something that simple for an older person, especially who's homebound and especially during COVID, it becomes really important. I can guarantee that health problems associated with diabetes are going up tremendously during COVID because people are stress eating People are not getting out of their house and exercising. People are watching more television. So I know this is going to show up as major health problems over the next few years as a result. Any other questions about this story? This is a really important story to understand.
Yes,
because I can
ma'am.
guarantee an essay question about this too.
So my question was, when you have type 2 diabetes, and I have some relatives that were with a proper meal plan and with exercise, then doctor was saying that that can go away. Is it just a time factor for the most part on when those desensitized cells become pre-sensitized? Like once you take away that overload of all the simple starch or simple sugars, when you stop flooding yourself with all that, as time passes, does that kind of bring things back into the normal rhythm? Is that what makes it go away? Is it not really?
Yeah, no, unfortunately, I think most of what they're finding, and I could be wrong in this, but my understanding, and I'm definitely not the expert on this, but my understanding is once the damage is done, the damage is done. But the way you control it then is by just limiting your carb intake. So, and eating the right carbs and eating them in small doses throughout the day. So, so a diabetic now just really needs to forever control what they eat and exercise. Otherwise, they will be on insulin and they will be on other drugs to control that. There are other drugs you can take, you know, without having to do insulin injections every day. There are drugs they can put you on at the beginning to kind of get that under control. But really, once the damage is done, my understanding is really for the rest of your life, you are on that controlled diet. Otherwise, it's going to get out of control again. And, you know, my grandmother finally, you know, a couple of years ago died at I can't remember now, was she 95, 96, something like that. And she had type 2 diabetes most of her adult life because of poor diet. And, you know, I'd go over and she'd have a box of donuts on the table and she was eating them and she didn't care. She's like, you know, I'll eat healthy tomorrow. So, you know, there are some diabetics who they'll have a day where they just eat what they want and they'll just deal with the consequences. But in general, you know, they just, for the rest of their lives, that damage is done. They can't ever get back to normal unless they just forever control their diet. And I always thought, you know, I had my DNA testing done recently, and I always thought I was genetically predisposed because both sides of my family, high rates of type 2 diabetes. But, you know, one side of my family all lives in Arkansas. And sorry to my Arkansas relatives, if you ever happen to see this video, no offense to you, but you eat poorly, you're fat, and you have diabetes, you know? So, and same on the, you know, my mom's side of the family mostly lives in California, but they're all really overweight also and have poor diets. But I just thought, for sure it's going to show up. Well, no, all my genetic tests showed that I'm not genetically predisposed. So all of those health problems on both sides of the family are 100% related to their
what
lifestyle.
they're doing to themselves, right?
Their poor diet. Yeah. I mean, in the South, you can't find a vegetable to save your life, you know, unless it's deep fried. And that's just how they eat. You know, you cook the eggs in bacon fat and then you scoop on some more bacon fat once they're done. And, you know, what is a vegetable? I don't know. We deep fry okra. We deep fry green tomatoes, but that is the only green thing that anyone ever eats. So much diet and lack of exercise for sure. High blood pressure, that's a really great question. High blood pressure is related to diet, but it's also hugely genetic, and cholesterol is hugely genetic. The way your body regulates cholesterol is hugely genetic. And we'll talk about that later in the semester too, for sure. So yes, it just becomes more maintenance, Denise. That's a really good way of putting it for the type 2 diabetes. You're trying to maintain normal, but you will never get back to cells that function normally again. You just have to be careful forever once that damage is done. Now that's different than, when we start talking about the cell, and this is the last thing I'll say because I know we're out of time. But when we talk about the cell and we talk about things like, how when you drink over time you become desensitized, like you can drink more alcohol without feeling it. But then if you don't drink for a while, you kind of go back to normal again. When we start talking about the cell, you'll see how that happens. That's different than this. Alcohol tolerance and drug tolerance and things like that, you can get back to normal in your cell because that's a different mechanism that's working there. Awesome questions today, everyone. Thank you so much. If you want to stick around and talk about lab partners, what I'll do is everybody else can leave. If you want to talk about lab partners, stick around. I'll say a few things to you and then you can do your thing, and I'm going to leave the room while you guys figure out the lab partner thing. So everyone else, thank you so much. I am going to stop recording for the lab partner part of this. So I'm going to stop recording now. And I do have an office hour this afternoon still from 3 to 4. So stop by if you have lab questions or anything else.